I'm Lily Simon, and this video is my 1.2 assessment, and we'll be looking at the process of muscle contractions and briefly describing the different types of muscle contractions. So firstly, we'll be looking at the overview of a muscle contraction, and it all starts within the nerve transmission, within the central nervous system and the peripheral nervous system. And that is followed by the neuromuscular junction, which is also then followed by the sliding filament theory. And the sliding filament theory is the process of how muscles contract to produce a force. In summary of this, the actin and myosin filaments within the sarcomeres, muscle fibers, binds together to create a cross bridge which enables them to slide past one another, which creates the contraction. Now I'm going to discuss and talk about how the nerve impulses are transmitted throughout the body. So here we start with the stimulus, which is a change in the environment, and in this case I'm going to use an example of a rugby ball coming towards a player in a match. So here, in this case, the receptors are the player's eyes, and the stimulus is the rugby ball. So therefore, the player is going to want to catch and receive this rugby ball. So a nerve impulse is a signal that is transmitted along a nerve fibre. It consists of a wave of an electrical depolarization that reverses the potential difference across the nerve cell membranes. So this stimulus is detected by a receptor, and in this case it will be the player's eyes, seeing the approaching ball that is coming towards them. These receptors will then send an electrical impulse through the sensory neuron. This will tell the brain that the rugby ball is coming towards them. So that is the message being sent to the brain. The central nervous system, which consists of the brain and the spinal cord, will then send a new impulse that travels through the motor neuron to tell it what muscles they want to contract or relax. The effector will then receive this particular impulse, then respond to what the central nervous system is telling it to do. Lastly, the effector will control the response, and in this particular example, the rugby player will want to catch the rugby ball. To enable this to happen, they need to contract their bicep brachii and receive and bring the ball in towards their body. Now we're going to be talking about the neuromuscular junction. A neuromuscular junction is a chemical synapse that is formed by the contact between a motor neuron and a muscle fibre. This is where the central nervous system receives an impulse and then sends a further impulse down the motor neuron. Once the impulse has reached the synapse, they chemically release a neurotransmitter which is called ACH, or also known as acetylcholine. This chemical then diffuses along the synapse and binds the receptor sites on the muscle, and once there is enough ACH that has been diffused across these receptor sites, then this impulse can now continue down the muscle. The impulse travels from the junction to the muscle by T-tubules. This impulse causes calcium ions to be released. And at rest, the actin binding site is covered by two different types of proteins called tropomyosin and troponin. Here this just shows the process of how this happens. So here the CNS receives an impulse, and then the CNS then sends a new one, and this impulse passes the synapse and the ACH is then released. This ACH then travels through the synapse and binds to the receptor sites onto the muscle. The impulse will then be able to continue down the rest of the muscle. So once the impulse has travelled across the synapse and reached the muscle fibre, it can then contract. So this just shows the different size comparison throughout this process. So it starts with the muscle, then inside the muscle are fascicles, and within them are muscle fibres. and inside those muscle fibres are myofibrils, within them are the sarcomeres, and smaller and within them are the myofilaments, and then finally they reach to the proteins, actin and myosin. We can now discuss and talk about a sarcomere. So this is what a sarcomere looks like. So these sarcomere are adjacent to one another, and they consist of alternating thick filaments called myosin and thin filaments called actin. This is also the reason why skeletal muscles look striated in appearance under a microscope. So now we can look at muscles at rest. So in this image you can see that the thin filaments are called actin and they have a binding site. And also the troponin is more globular, whereas the tropomyosin is a long thin strand. So this image shows a thin twisted protein which is specific to the active sites for myosin to bind to. Myosin is a thick filament with a tail and a globular head, and that attaches to the actin active sites during the contraction. So now we're going to look at what happens before the contraction. So the impulse travels through the cell membrane along the muscle fibres. The impulse reaches a sarcoplasmic reticulum. The calcium ions are then released, and these calcium ions can then bind to the troponin. Troponin and tropomyosin both retract away from this actin binding site, which allows myosin to attach to the binding site. So in this photo, we can now see what happens during the muscle contraction. So here, the actin, which is energised by the ATP, causes a power stroke, which is the movement of the sarcomeres being pulled closer together. The Z lines, which are here, are pulled together, which is known as a power stroke, which creates a contraction, and that pull onto the muscles. So therefore the sarcomeres are getting shortened as the muscle contracts, as the Z lines are coming closer. So these two lines here come closer to the M line, which causes the contraction. And on this side, it shows what happens before the contraction, and this is during the contraction. So the I bands get smaller. And here is just a summary of this. So the actin causes the power stroke, the Z lines are then pulled closer together, and then lastly the sarcomeres are shortened by the contraction. Now we're going to move subject on to muscle contractions. So now we're just going to talk about the end contraction again. So the myosin arms need more ATP in order to detach from the actin binding sites and to relax. The calcium ions are not released once the impulse has stopped. So troponin and tropomyosin return to their original shape and then cover the actin binding site back up. Okay, now we're going to move subject onto the different types of muscle contractions. So there are 4 different types of muscle contractions, such as concentric, eccentric, isotonic, and isokinetic. Eccentric and concentric are both forms of isotonic muscle contractions. This means that they change length when they contract. So firstly, we're going to talk about isotonic concentric contractions. So in a concentric contraction, the muscle tension rises to meet the resistance, then remains the same as the muscle shorten. So here is a picture of an example, and this is just a man doing a 25 kilogram bicep curl. So a concentric contraction are those which cause the muscle to shorten as it contracts. An example is bending the elbow from straight to fully flexed, causing a concentric contraction of the bicep brachii muscle. Concentric contractions are also the most common type of muscle contraction and occur frequently in daily and sporting activities. Now, the next muscle contraction is isotonic eccentric contraction. So an isotonic contraction maintains constant tension in the muscle as the muscle changes length. An isotonic muscle contraction can either be concentric or eccentric. And this is another example photo where the muscle contracts but does not shorten. Now, isometric contraction, or also known as a static exercise, is one which the muscle fibers, where the muscle fires but there is no muscle movement at the joint. In other words, the joint is static but there is no lengthening or contracting. So when the muscle contracts, there basically is no result in movement. And here is another photo where the muscle just stays the same length. And the last muscle contraction is called isokinetic contraction. So an isokinetic muscle contraction is one in which the muscle contracts and shortens at the constant and consistent rate of speed. The main characteristic of an isokinetic muscle contraction is that they have a constant speed. So there is no change in length of the muscle, and there is also no movement at any of the joints. But the muscle fibers still fire. A solid example of an isometric exercise includes pushing hard against a wall. This is where the muscles are still being utilised and potentially being stressed, but there is no movement at the joints. Also, dynamic constant external resistance, or also known as DCER, is used to experiment isokinetic contractions because it better describes the notion that resistance remains constant but not muscle tone when lifting weights. So this is where they use a machinery, you're strapped into machinery and you create a muscular force by your leg, and the machinery will copy that exact force so there is no movement in it. So this now concludes my 1.2 muscle contractions assessment video, so thank you very much for watching.